All right. Hi, everyone. It's Stephanie with the Patient Story, and I am so excited about today's event. I'm so glad that everyone could join us today. We are talking about treatment options for relapsed refractory multiple myeloma patients. And of course, this is for everyone, people who are diagnosed with myeloma, and of course, the caregivers, the unsung heroes in all of this. And I want to stress that today's event is different from maybe other webinars that people have been a part of. We're going to try to take medical jargon out of this as much as possible. Of course, it's a part of the conversation, but this is about humanizing what this means for everyone out there, because as we know, there are so many things to talk about in this space. First, I do want to say thanks to Karyopharm for their support of our educational program today. Now, we will be asking our doctors today questions that have been generated also from the audience, but anything that doesn't get answered today, we will try to do some follow-up after. And I want to kick off today with an introduction of our special guests, our top myeloma specialists today, Dr. Krina Patel from MD Anderson and Dr. Mohammed Balyevich from University of Nebraska Medical Center. Welcome.
Thank you for having me.
As we go into this discussion, again, we have a lot of ground to cover. I'd like for you two to set the stage a bit, because in terms of multiple myeloma, it's unfortunately not curable, treatable, and especially with more and more FDA-approved treatments that are coming out constantly, it feels like. But I think it's important to establish that we have different kinds of patients here. You've got, the early relapse, the late relapse, the standard risk, high risk. I mean, there are a lot of different buckets. So for this general discussion, I think we'll be covering more of, say, the after a few lines of therapy type of patients. But how would you like to, categorize the different buckets in terms of when you look at a patient, how you decide which, bucket to go to first in terms of treatment paths? And we'll start with you, Dr. Balyevich.
Thank you. That's a really good question and important question for us in general. Multiple myeloma, as you point out, unfortunately, as of yet, still is uncurable malignancy. I will add that hopefully every decade brings us closer to the cure, and it's a lifetime worth spending in search of it, I would say. But you're absolutely right. There's many different things that we consider when we are looking at patients with multiple myeloma, particularly, as their disease relapses, which in vast majority of patients, unfortunately, it does. I would say that we all have probably N of one or N of few, where patients may be in a very, very long, durable responses. And some of them actually might even be off of all therapy, but it's important to know that those are exceptions and not the rules. So when it comes to understanding of how we approach relapse and or refractory myeloma, different things play part, really. Duration of initial response, defines biology. In general, we like to think that drug combinations are better than fewer drug combinations. So, for example, triplets rather than doublets, or in the newer times, even 4 drug combinations rather than fewer than that. When it comes to prior drug exposure and refractory status or toxicity with prior drugs, that's usually something that helps us, decide specific drug and combinations that we might want to move forward with. Age, frailty, or comorbidity of patient preference, and their goals of care, as well as the logistics of drug administration. So is it oral, is it IV, is it subcutaneous? Helps us decide, dose and schedules of treatment. And then when we talk about the high risk versus standard risk, that's also extremely important. And also whether or not they're transplant eligible, have they received transplant, etc., helps us decide oral treatment approach and goals. When it comes to prior exposures, we like to pay attention to any presence of peripheral neuropathy, for example, in which case we might want to avoid drugs like bortezomib, any cardiac or heart, comorbidities or diseases, or failure, in which case we like to avoid drugs like carfilzomib or anthracyclines. Whether or not they have renal insufficiency or impaired renal function, in which case we often need to adjust dosing of some of myeloma medicines, such as lenalidomide, whether or not they have had reactions to these drugs like rashes, etc. And in case those were severe, then that may really make us hesitate in terms of choosing some of the newer examples within those classes. So, and lastly, what's important to note, of course, as we live currently in the unprecedented times of pandemic, right? As we mentioned before, the modes of administration, the frequency of visits and monitorings, et cetera, we have to recognize that all oral regimens, are more convenient and can be delivered for patients, while they take their medications at home. But, compliance needs to be monitored. As we mentioned, IV infusions, they do require a clinic visit, but there's a question about a frequency of administration, which is an important factor, and also some medications having a subcutaneous version that's available instead of the intravenous one. And lastly, of course, we have to monitor any potential toxicities, including hematologic toxicities or any other intolerances that can come in, particularly with some of the newer agents. And all of this put together really gives us a broader picture in terms of how we approach every single patient individually and what decisions we may make for early relapse and particularly late relapse.
Well, thank you for summing that all up. I know it was a lot of ground to cover. And by the way, we will be delving into side effects and management later on in the conversation, because I know that's obviously so important for people. Dr. Patel, do you have anything you want to add to that? And also, if you want to segue into how and when you decide to restart treatment for a patient, what are the factors there? Examples are, going from MRD negative to positive, or symptoms, or M proteins rising to a certain level. I'll
Sure.
have the floor.
Thank you. I think Dr. Baljovic did a great job of telling you all the different, things that we look at for each patient to make sure we have the best outcome for that patient that's in front of us. To make it a little simpler and summarize, I think when my patients come to me, I saw 25 patients yesterday, and each patient, I don't get very much time with all of them. But with each patient, we see how they're doing. And really our goals are, for most patients, I think, and most people in general, goals are, you want to live longer and we want to feel well while we're living longer, right? We want good quality of life. And so that's really why we look at all of that. So for my patients, when they come in, we look if they're high risk or standard risk, because that can change what I look at in terms of the M protein or MRD, et cetera. It can be defined differently and it can mean different things for high risk versus standard risk patients. And then I look at patients to see if they're frail or fit. And if they're frail because of the myeloma, that's different than if they're frail because of the comorbidities Dr. Baljovic just talked about, right? And so really that's how we decide on all the different treatments that are available. What's the best next step? I think the guiding principle is that the earlier you are in treatment, no matter what line of treatment it is, but the earlier you are, we have more options because your myeloma is not as resistant. So our goal is to give you those best therapies up front. And then we kind of use other things down the road. But as new research comes in, we actually get better therapies down the road. And that's the nice thing about myeloma, that we have so many new things coming in all the time that really you can actually get better responses later than something that maybe didn't work as well earlier because we have so many options. So I think, we have these guiding principles, but things are constantly changing. That's the other thing to think about. And so in terms of when do we change therapy, right? So of course we have the IMWG criteria that everybody uses, but in application, if I have a patient that's really slow growing myeloma that, is doing really well on their therapy and their M proteins going up by 0.1 every 6 months, I'm probably going to watch that patient until their M protein gets to 1 maybe, instead of the 0.5 that we usually say is a biochemical relapse. So if someone gets to 0, they get to 0.5, that's biochemical progression, as long as they don't have any of the CRAB criteria, so the calcium, the kidneys, the anemia, the bones, right? So if they're really slow growing and I know they've done really well for a long time on other therapies, I'll just watch it. And then once they get to 1, maybe I'll say, okay, let's go ahead and switch therapy now. Versus my high risk patients, when I start seeing that M protein start going up, I'm watching them very closely. And the second they get to 0.5, which could be in a month, maybe 2 months, we switch therapy. And really, it's about the standard of care therapies that are available versus research trials that are available. And in the end, standard risk, high risk, we want to get you to the best therapy we think is there for you. But my higher risk patients, I tend to do more research trials earlier because we know the therapies we have already still don't do as well as they do for standard risk patients.
Much like what Dr. Patel said, in my clinic, I have 2 goals. One goal is to try to prevent myeloma from shortening people's lives. And if left unchecked and untreated, that's invariably what's going to happen. For some patients, sadly, very rapidly. For some later, but invariably, people don't live with hematologic malignancies, myeloma or other, without major effect on their livelihoods. And two, while we do anything that we decide together with patients, what we will do, that we do it in a way that really allows them good quality of life, and that it meets their goals of care, and that we respect their idea of what it means to have a cancer and what they wish to pursue in terms of our best efforts to suppress it, eradicate it if we can. As we said, very challenging with myeloma, but at least, having patients be in a position where their disease is not dangerously influencing their life, or as we said, shortening their life, while at the same time allowing them to live it with a good quality and with dignity. There's many other things, quite frankly, to consider. I think that the principles Dr. Patel explained in terms of high risk, standard risk are really quite important, very, very important. And the role of clinical trials and participation in clinical trials is, of major value, not just in myeloma, but in any cancer. And quite frankly, as I started sort of my work as an independent physician, some years ago, the analysis that I took a look at when it comes to myeloma and other cancers, by the mechanism of action, the new regulatory approvals that we have seen in myeloma over the last number of years is really only lung cancer is rivaling that. So we are very, very busy, and that's good, conducting new clinical trials and trying to find solutions that bring better answers for all these phases of disease and all these situations. And that's really a discussion and a conversation that we always have to have with our patients.
I love how both of you have talked about access to this care, the newer treatments, and also quality of life, because we hear it, talked about, but when you talk to patients, obviously, you really understand the impacts to day-to-day living, and that's a huge consideration. It's, yes, I want to extend my life, but I also want to be able to live it to the best of my ability. So, Dr. Baljovic, you talked about, these different mechanisms of action, and we've started to see some newer ones being introduced and approved. So let's go ahead and start talking about some of these. And I'd like to start with selinexor, which I know has, at first it was for patients who had at least 4 previous treatments, 4 lines of therapy. And then now with the bortezomib and dexamethasone is at least 1 prior line of therapy, which of course means very different situations and you're gonna have different experiences. But let's open it up to you explaining the mechanism of action, like how it works in layman's terms, and anything else about what you've seen with selinexor.
Yes, absolutely. So selinexor is just one in the line of new agents that have entered the multiple myeloma treatment space. Selinexor, or XPOVIO, is a first-in-class oral selective inhibitor of nuclear export. So in layman terms, what that really means is, if we just take a step back and look at how our bodies are made, they're made of cells, right? And each cell has a membrane, has a sort of internal compartment. The liquid compartment has a nucleus where all the genes are, et cetera, where major processes are happening. So there are pores in the nuclear pore that allow transitioning of various proteins and molecules inside of the nucleus and outside. And what selinexor does is it is a selective inhibitor of one of those nuclear pores, exportin 1 or XPO1, which is a major nuclear export protein for a variety of really essential proteins in the biology and the life cycle of any cell. So that includes tumor suppressor proteins, that includes proteins that are called oncoproteins, which are in general terms proteins that stimulate division and may cause the cancer to happen. We know that this XPO1 is expressed in myeloma, and the high levels of XPO1 levels enable escape from these tumor suppressor protein mediated cell cycle arrests and apoptosis, which is a process by which cell dies. And we also know that the XPO1 levels correlate with poor prognosis and drug resistance in multiple myeloma. So then when we look at what selinexor really does for patients and against their myeloma disease, is it reactivates multiple tumor suppressor proteins by preventing nuclear export. It inhibits oncoprotein, meaning the bad protein that we mentioned earlier, translation, meaning getting it to the active phase. And then it also works synergistically, meaning it helps, right? It works with the glucocorticoid or steroid receptor signaling in the presence of dexamethasone. So what you alluded to earlier, selinexor was approved several years ago, initially following the STORM trial, which described the selinexor use in twice weekly setting, which, truth be told, was very, very challenging and resulted in multiple side effects, namely GI side effects, from the standpoint of symptoms, and also some, side effects in terms of lower counts and myeloid suppression that normally physicians follow for their patients. However, since then, we just had recently published data from the Boston phase 3 trial that looked at combining selinexor with bortezomib and steroids and compared that to the treatment with bortezomib and steroids alone. And that phase 3 trial showed a very meaningful progression-free survival benefit with this triplet combination versus the doublet combination. But one of the most important things that trial taught us and showed us is the fact that we can use selinexor on a once weekly schedule rather than twice weekly schedule. And in fact, some of the follow-up studies, including the STOMP study, which is a multi-arm, more than 10 arms of different selinexor combinations with different modern classes of drugs like carfilzomib, which is a newer cousin of bortezomib, or pomalidomide, or even some of the newer drugs that we'll get to probably very soon. So all of these arms, and this trial is also demonstrating that, in fact, use of selinexor on a weekly schedule is first of all, safe and feasible, it appears to be, and it's also fairly effective. So I look at selinexor maybe as a drug that's been described by some as a horse that stumbled out of the gate. And now we are learning, and in fact, that's a whole history of myeloma, quite frankly. If you look at what we've done in the history of this malignancy, we have learned we initially used very high dose steroids, and now we learned not to do that. We have used bortezomib as an intravenous drug, now we use it as subcutaneous. Earlier immunomodulatory drugs, we used very high doses of thalidomide, but now we have lenalidomide and pomalidomide. And these even in daratumumab, our antibodies, we used to use it intravenously, and now we have subcutaneous form. So we always learn, right? And we are learning rapidly with selinexor. And I think it's a very important drug, in my opinion, because it offers, first of all, novel mechanism of action that's very important for our patients, because one of the cornerstone principles of treatment of myeloma is the fact that this is disease that generally benefits from combinational therapy rather than single agent therapy. And that's not to say that, always adding more is better. There is a certain threshold at which you start, observing more toxicities rather than gaining benefit. But the principle of long-term management of myeloma really asks of us to have newer drugs with newer mechanisms, the ones that myeloma cells have not seen, the ones that they may presumably not be resistant to, that they may be more sensitive to, and selinexor definitely offers that. And as we continue pursuing some of the additional studies, as I mentioned, we are going to continue learning even better ways of utilizing drug like selinexor. And in my clinic, I do depend on it for patients with multiply relapse refractory myeloma. I also think it's very important, and we will hopefully, provide some educational materials for the viewers as well, to look at how we manage and prevent as much as we can side effects that can be a result of selinexor treatment. So that, as we mentioned, is GI toxicities, nausea, vomiting, diarrhea, and also cytopenias, reductions in the counts. So whenever I use selinexor, either as a standard regimen, I like to use it, obviously, as we said, on a weekly basis, and usually in combination with some of these newer generation proteasome inhibitors or immunomodulatory drugs like carfilzomib or pomalidomide. The arm with the daratumumab combination has already been published and showed efficacy. I like to use it with a proactive stance towards management of the side effects. So this is not a drug that we give to patients and tell them just go home and come back in a month. we check in on them. We ask them to be proactive about letting us know how they feel. I usually provide prescriptions to multiple of these supportive care medications, such as Zofran, of course, always needs to be part of antiemetic pre-medication. We always ask patients to take dexamethasone before their selinexor because it helps. Olanzapine is very commonly something that I give and will ask patients to use in general. And sometimes I may even use growth factor supports for even white cells or platelets if we need to try to support or prevent severe reductions in counts, which would otherwise either prevent us from treating patients or make us reduce the doses, which would then compromise on the efficacy. So all of these are important things when we are thinking about. And last thing I will mention about selinexor is what I think is also exciting is that it's not associated with, as far as we know so far, obviously it's a new drug, but it's been on the market and it's been in investigation for a number of years. As far as we know, it's not associated with risk of secondary malignancies, with the risk of rashes, for example, and may potentially lend themselves as an agent of choice as we start tackling progressively more and more some of these pre-malignant conditions, with any of myeloma agents. So when you have an agent that, doesn't risk, like, for example, alkylating agents or even immunomodulatory drugs are known and associated with some of these risks, that may potentially be useful.
Yeah, no, I think that was perfect. I think the biggest learning points are that, lower dose, the once weekly dose works a lot better. Proactively giving 2 antiemetics or 2 nausea medicines ahead of time and potentially even a third one. Most of my patients get fluids once a week, at least the first month, to make sure that they don't get dehydrated, especially in the summer in Texas right now. So we kind of do a lot of proactive things to make sure that they do well without those side effects. And then it's monitoring. And usually after that first month, people start doing better with it, even if they were having side effects. So that's what's a little different too, is that unlike other medications where we start to see side effects long-term, here, those GI side effects tend to actually improve, which is good. But, we want to make sure people's weights are doing okay, et cetera. So it is a little bit more monitoring that first 4 weeks, but then it becomes a little bit more like clockwork. So.
We like to hear clockwork. Some sense of what to expect is always nice, I think. And wrapping this up too, I know it's given orally, and that's another big topic, right? And Dr. Baljevic had talked about it, how there have been iterations too on whether it's IV to subcutaneous, like a shot, or oral, a pill. And I've talked to many patients, of course, who are like, yes, please, I want the pill, like, give me that. We have 3 more of these to get through, and then I'd love to be able to get to, the CAR T and the bites. So let's go ahead and go to melflufen, which has been FDA approved with dexamethasone for relapse refractory patients with 4 more lines of therapy. Dr. Patel, actually, if you want to just sort of describe that, that'd be great.
Sure. So it's sort of, melphalan is the drug we've had the longest for myeloma besides steroids. And here you're actually repackaging it to be bound to peptide that helps it get, once it gets into the myeloma cell, these aminopeptidases that are in the myeloma cell help break it down so that then the melphalan can actually do what it does, which is cut the DNA like a regular alkylator like melphalan does. And that when you cut it, it can't survive. And so it's using an old drug, but getting it to the myeloma cell, right? So you're actually targeting it into that myeloma cell compared to regular melphalan. When you get it IV, it sort of goes everywhere and it goes into all the cells that are dividing faster in your body. So toxicity tends to be higher. That's really the thought process, that here you get more melphalan concentrated into the myeloma cell, activated in there, which kills more myeloma cells and hopefully gives you less toxicity. And in general, it has actually been very well tolerated for patients. And so once again, melflufen plus dex, it's considered a doublet. So likely not the best regimen, but I do think for patients, especially my older patients who are more frail, who couldn't go through stem cell transplant or couldn't really get alkylators, I think it's a great substitute for alkylator therapy in myeloma. And I know there's been other trials that have been going on. There have been some safety issues. So I think just like all other therapies in myeloma, we have to kind of let that play out first and really see which patients that it's probably the best for. But I do have a patient population that I think, really having a more targeted alkylator would be beneficial for.
Okay, thank you. And anything, I know you said generally tolerated pretty well, but anything you want to point out in terms of side effects and management that you tell your patients?
So I think, counts are one of the big things with alkylators in general. So platelets and white count. So infections in case your white count's low, et cetera, neutropenic fever. And then their GI toxicity actually wasn't so bad. You would expect with melphalan to have really bad diarrhea, nausea, vomiting, and it was actually pretty well controlled. So nothing else that I can really think of that was out of the ordinary.
Thank you so much, Dr. Patel. We're going to switch over to isatuximab now. And Dr. Balievich, we know that it's been approved for various situations. So with pomalidomide and dex for patients with at least 2 previous therapies, and then with carfilzomib and dex for patients who've received between 1 and 3, right? So I just wanted to get that out of the way. What would you like to say about isatuximab and how it works and when you decide to prescribe it?
Of course. So as you mentioned, isatuximab is another example of a monoclonal antibody that targets the CD38 receptor on the surface of the myeloma cells. It binds to this specific epitope of this CD38 protein molecule, resulting in anti-myeloma effects through several mechanisms, which include antibody-dependent cellular-mediated cytotoxicity, complement-dependent cytotoxicity, antibody-dependent cellular phagocytosis, and direct induction of apoptosis mechanism similar to those reported for other monoclonal antibodies. So this was really a mouthful, but basically various different ways of engaging immunity and causing death and toxicity to the myeloma cells, either directly as an antibody or by sort of calling your colleague helpers from the immune system to try to attack the myeloma cell. As you correctly mentioned, isatuximab was recently tested, and the results of the both ICARIA and IKEMA studies were presented, and as you also correctly pointed out, in different patient populations. I would, rather than go into really specifics of sort of the overall response rate and toxicities, et cetera, what I think is important to know is the following. A mounting amount of data, an increasing amount of data is suggesting that incorporating CD38 antibody directed treatments to myeloma patients is leading to benefits. The overall response rates and the length of time in which whatever the response may be in the end, which by and large is very, very deep, responses such as stringent complete responses and also significant proportion of MRD negative responses. So we know that incorporating CD38 targeted therapy with monoclonal antibodies is very good, and that it brings a huge benefit to treatment of myeloma. And this also brings up another question of sort of your best foot forward, right? When we want to try to treat patients, we always try to give them the best treatment when they are sort of the youngest, when they're the fittest, when they are least tired, if you want to say, from various treatments, when their bone marrows are least damaged over time, etc. So that's really important. 2, daratumumab was the first antibody that targets CD38 that was approved, and is an antibody that we have the most experience with. It's been on the market for far longer than isatuximab, but isatuximab is also coming up. So the 2 studies that you mentioned are examples of studies in relapsed refractory myeloma, either in combination with pomalidomide, with IKEMA, or with the carfilzomib, with ICARIA. And these are examples of neurodegeneration drugs that bring significant benefit to our patients with relapsed refractory disease. My last comment on sort of what this means is really to do with always knowing what the data is exactly telling you and showing you, and in which patient population was it studied. So for example, the isatuximab and pomalidomide didn't allow prior exposure to Pomalyst, right? So if you have somebody who's progressed on Pomalyst and refractory, maybe that might not be the best combination to think about. Similarly, isatuximab carfilzomib, a trial looked at patients who didn't previously get carfilzomib. So these high response rates are really contingent upon understanding who that benefit is for, and does that really apply to your patient? These are fabulous results. Isatuximab has been approved as a result of both of these studies and is probably going to increasingly be incorporated into the treatment paradigms. There are some major differences, for example, between these two antibodies. Daratumumab does have availability of subcutaneous dose, but perhaps the pricing of isatuximab appears to be a little bit more favorable to daratumumab. We need bigger studies to really prove this, but there's points about both of these. Pricing, the way you administer it, etc., that really need to be taken into consideration. And which one of these combinations would you use, isatuximab or daratumumab combination, etc., with any of these other agents? It really has to be individualized decision for every patient of yours. And sometimes, you will make a decision that may rely more on what the patient preferences are, and that's okay, as long as you, sort of educate your patient on what would be perhaps the most beneficial regimen in that exact moment for their specific disease.
I love that message. It's obviously tailoring to each person because this disease appears in so many different ways for so many different people. I will lob this one to Dr. Patel. The last one in this category I want to talk about is the belantamab mafodotin. Please forgive me if I butchered that, aka Blenrep. And so the FDA has approved it for patients with 4 or more prior lines of therapy. And I'm generalizing this to be broad strokes again. But Dr. Patel, if you could describe that mechanism of action, aka how it works, and when you decide to prescribe it.
Sure. So this is our first BCMA-directed therapy that we have for myeloma. So once we found BCMA, or B-cell maturation antigen, this is a flag that's on myeloma cells and not on any other major organs or tissues. It was a huge discovery. Basically, everybody started attaching anything they could to that BCMA to get it to that myeloma cell to kill it. So what we talk about next with CAR T and bispecific, same thing. We're using that target BCMA to get some type of drug or immune therapy, et cetera, to the myeloma to help kill. And so here you have something called an ADC, or an antibody drug conjugate. So you have the antibody for BCMA, and attached to it, you have this drug that I won't go too much detail about how that drug works, but basically when you get the infusion, so it's an IV, when you get the infusion, it's once every 3 weeks. It goes in and it actually attaches to the myeloma cell, kind of like the anti-CD38 antibodies do, but instead of CD38, it's BCMA. And now this drug gets put into the actual myeloma cell. So kind of similar to that melflufen story, it just works a little bit differently of how it gets into the cell. But once it gets in there, it actually kills the myeloma cell. And so the early studies, once again, were doublets. So Blenrep by itself, and that's what's approved in 4 lines of therapy. The response rates are very similar to single agent daratumumab, so in the 30% range. And my patients have actually done really well on it when we've used it. And I tend to use it for, my patients who might have a little bit more frailty or, are older, and I really want to give them just a single agent that they're going to do better with that. And I know that they have a good chance of response. Those are my patients that I really put on there, initially. The nice thing about it is that they have done studies in patients with kidney problems, so kidney damage, which a lot of our therapies, unfortunately, we can't use safely in patients with kidney problems, or we have to be really careful when we use it. And their studies actually showed that even patients with, 50% reduced renal function, their kidneys weren't working as well, they still got great benefit from it. And so I've actually used it in a couple of my dialysis patients recently and had some fantastic responses without major side effects. So, those are things that we're all sort of learning now as we try to, use these drugs. But I think it really, that group of patients is one of the unmet needs for myeloma. And that's where I really like seeing what I see with Blenrep right now. Having said that, the big question really is BCMA therapy. can you use different ones? Can you use them in different order? If you use one, will it make another one in the future not work? So because of those types of questions right now, we don't have answers for that. So we're a little bit more careful, and instead of giving everybody Blenrep, the other part of it is it's also a high maintenance drug compared to similarly to what we've talked about already today, in the sense that it can cause eye issues. So it can cause these little microcysts in the eye that we call keratopathy. And for most patients, they don't have symptoms. But if it gets really bad, it can cause ulcers and all kinds of things that you don't want. And so you have to see an ophthalmologist for an eye exam, or optometrist for an eye exam every single time you're about to get the drug. So that's why I call it high maintenance, because you have to see me and you have to see the eye doctor. And so, we're learning more about it once again, and patients are doing better. They're looking at, giving it every 4 to 6 weeks instead, and patients still do just as well and maybe have less side effects. So those are all the things coming down the line. They're putting it with everything, all the different therapies out there for myeloma, which will likely increase that response rate and doing it sooner. So I think there's a lot of exciting things coming down the road with it as well in combination. But even as single agent, it's our first BCMA therapy, and it seems to be pretty good for at least a subset of our patients.
It's great to know that, especially for the unmet needs category of people, that there are options that are coming out, right? So it may not fit the bill for everyone. And I also appreciate you bringing up the keratopathy, the issues with the eyes, because that is the most talked about side effect when it comes to Blenrep. And that's why if you as a patient or caregiver hear about the risk evaluation and mitigation strategy, or REMS, Dr. Patel really spelled out for you what that really means, which is your oncologist or hematologist, you're going to be working with the ophthalmologist or optometrist to make sure there's constant communication about what's going on with your body. So thank you so much for that. As we wrap this part, is there anything else you want to talk about with side effects? I know that both of you have addressed the nausea and vomiting and the antiemetic sort of regimens that you like to give patients, and not to chase, right? To give them beforehand, and by the time you're feeling the nausea, it usually is pretty hard to, catch up with. And you've also talked about anemia, I think. Anything about neutropenia or loss of or decreased appetite? Any other GI issues that you want to address in terms of how you advise your patients to, minimize those toxicities?
Well, I want to really echo everything that Dr. Patel said about, Blenrep, and just reinforce, what she mentioned as well, which is the fact that we are hopefully going to learn how to maybe optimize the use of Blenrep. We talked about this before, selinexor, but many of us believe that perhaps dosing of, belantamab can be stretched out a little bit without compromising the efficacy too much. And with these issues with corneal, toxicities and such, it can be really a big problem. And, unfortunately, another thing to raise is we don't have good treatments for this. The steroid drops were initially part of sort of supportive measures, but really they don't prevent or dramatically help. We do advise all our patients to use artificial sort of moisturizing solutions for their eyes, etc. But it's really the sort of very, very proactive management that we have to employ with Blenrep. And as Dr. Patel mentioned, what makes it easier is that it can be either ophthalmologist or optometrist, as long as somebody looks. And this is all done through this so-called REMS program that both, all providers need to, be registered through in order to administer this therapy. So I think, another example, as we said, of a drug that we may potentially learn in the near future that could be used in slightly different ways, that can optimize the entire experience for patients. In terms of your question for neutropenia specifically, we talked about the monoclonal antibodies in some of these combination trials. Neutropenias have been and various other cytopenias, but neutropenias in general as well have been showcased as a toxicity. And, that just requires really proactive management and following. And any myeloma treatment, in fact, can cause neutropenia or anemia or reduction in platelet counts. And, it's a combination of measures of proactive management, dose reductions, sometimes the growth factor support, that are required in order for you to achieve the sort of the most optimal end result. I will also mention lastly, approach is not the same in every single patient. We all have patients whose bone marrow is completely filled with myeloma, unfortunately, and then their counts are low as a function of impaired bone marrow function, which normally produces counts. So, sometimes you just have to treat through that with supportive transfusions and supportive medications to try to clear out the bone marrow and try to allow it to work better. And that's scary sometimes, to try to treat through those types of difficulties, but it is sometimes necessary. So our approaches really do vary and can change from situations where we like to hold off, wait until the kind of recovery, maybe support some, to all the way to you got to treat through it in order to, help the patient. So it's, combination of different strategies.
Absolutely. Not one size fits all. And you're both highlighting why specialists are so important, because you have so much coming out and you need to have someone who, for the most part, especially when you get to more complicated situations, who really knows all the different options. Now, I'd love to switch over to the T-cell therapies, which are really exciting with both CAR-T and the bispecific T-cell engagers, or BiTEs. So Dr. Patel, I'll let you take it away.
Okay, so I promise this is not going to be lecture style. It's really more for teaching and just to understand how these sort of neuroimmune therapies work. And specifically, I'll talk about CAR T cells and the bispecifics, which are, some of the biggest things that are happening at all of our big hematology conferences and oncology conferences, and on all the patient support, social media, et cetera. I get things all the time from my patients that they found on, different patient support channels that they have about CAR-T and bispecifics. So what is a CAR-T? So T cells are a type of white blood cells, a type of lymphocyte we all have in our body. And you've probably seen, the white blood cell called lymphocytes in your blood test, and T cells are one of them. And they're pretty amazing. So remember I told you I had gotten a C in immunology in med school. So I learned a lot during residency and fellowship to do this. But T cells are sort of our army men, right? I think of them as troops, as soldiers, and they have been born with a different receptor. And I call that night vision. So when you have these different T cells that are supposed to fight fungus and viruses and all these things in the world that can harm us, those T cells, they have a specific lock and key with those different pathogens. And what we can do now is we can collect those T cells by blood, almost like a dialysis session. It's a one-time, 2 to 3 hour session where we put a line in, take your blood out, put the rest of the blood in, take your T cells out of there. And then we can put a new receptor in there, this new night vision, right? So it's on BCMA, that B cell maturation antigen again. So that goes into the cell through something called a viral vector. And so now every T cell that we grow in the lab specifically is made for BCMA. And so that's grown, and there's all these other things that help the T cell when it finds that myeloma, it helps it grow better and kill better. So all these things like CD8 linkers and 4-1BB, et cetera, it's a lot more detailed than you need to know, but really we're giving your T cells something to go find that myeloma, right? And so the two that have been talked about the most are ide-cel and cilta-cel. And ide-cel is the one that's just recently approved as standard of care. And there's a couple of differences between it, but really, I'll show it here. The number of cells you need is a little bit different because of the way the CAR-T is made. But for ide-cel, we usually try to get to 450 million cells. And in cilta-cel, it's about 50 million cells because it's per kilogram. So it's just dosed a little bit differently. But both of the big trials for this had, patients who had 6 lines of therapy. So they had pretty much everything already that was available to them. And when we say triple class refractory or pentaclass refractory, this means the different classes of myeloma drugs. So triple class is proteasome inhibitors, IMiDs, and CD38 antibodies. And then penta-refractory is that you've had all the proteasome inhibitors, 2 of them, plus the 2 IMiDs, plus the CD38 antibodies. So these patients, were refractory, meaning that they had already relapsed while on these therapies. So they had pretty bad disease. And then for the ide-cel, you actually had extramedullary disease, meaning that it's not just in the bone marrow and the bone, but you could have it sort of in soft tissue. And that disease is a little bit harder to treat. It's more aggressive. So, a lot of these patients had pretty aggressive disease, and a lot of them had a lot of myeloma going into it. So you really can't compare the two in terms of outcomes because the patients are different, but just to show that both had really, relapsed refractory patients that had a good amount of disease and high risk disease too. And those are patients that usually don't do well on clinical trials or even with single agent, therapy. But here, I mean, you see this response rate of 73%, 98%. I mean, mostly when patients were with daratumumab or Blenrep or selinexor, all these single agents, the response rate is 30% when they get approved as single agent. So this blew that out of the water, right? This is why I'm so excited about this, and I have conflict because I do research with CAR T, but this was huge. This was a great response rate in a patient population that had already had those other drugs, and really we didn't know what else we would be able to give them. And the deep responses, the CR responses, right? A third of patients in ide-cel and 80% in cilta-cel had these really, really good responses where we couldn't find the myeloma in the bone marrow anymore. And same thing with MRD, that minimal residual disease, looking at it to the millions, and we still couldn't find those myeloma cells. And then PFS is what I call the hibernation period for myeloma. And if you had that deep response, it was 19 months for ide-cel, and in cilta-cel, it looks like it's 22.8 months for all patients. So, pretty impressive one-time cell dose, no other therapy afterwards. So you're really just, coming in for blood count checks, things like that. And to have that long, 1.5 years to 2 years without needing any other therapy, that's a huge window that tells us so much about, new things that we've learned with myeloma. So I think that's where the excitement really comes from. And here, I'm not going to go too much into it, but it just showed that people who are older actually had great response rates. I'll move this over. They had great response rates. People who had high risk disease still had really good response rates. People who had extramedullary disease, and these triple refractory, penta-refractory. So even our high risk patients had some fantastic responses, which is exciting. And then the other thing for both ide-cel and the next slide for cilta-cel, really the patients who had those deepest responses, the CR or stringent CR, which is this light blue line, did way better, did a lot better for longer than patients who got a VGPR or PR. So from all of our therapies, we know that the deeper response you get, the better you do. So now our goal is, how do we make these patients that are down here come up here? So how can we improve the current therapy to make sure everybody gets that CR, stringent CR? And then how do we hopefully cure some of our patients? And even with the cilta-cel, the patients who got that stringent CR and that teal line, the green line, did better for longer than the folks who got a little bit less than stringent CR. So that deeper response really matters. So then quickly into the toxicity, because this is also completely different toxicity than we have for anything in myeloma. I think most people try to think of this and compare it to stem cell transplant, but it's not quite the same. It's a cell therapy, yes, but in transplant, you're using chemo. You're using that high dose melphalan to really just take everything out of the bone marrow, and you give the stem cells back for, normal immune cells again and platelets. Here, we're using those T cells to kill that myeloma. So what happens, it's like when people have the flu, right? Remember I told you the T cells are used to killing viruses and fungus, things like that. It usually gives us fevers, sweats, it can give us chills, it can make our blood pressure go up or down. And that's really what cytokine release is, that when these T cells engage and they kill that myeloma cell, they release all these cytokines that help other white blood cells come there to help with the fight. And really those cytokines then lead to fevers and just not feeling well, fatigue, et cetera. Even though there's a lot of it, 84% and 95% of patients that get CRS, these are all grade 1, 2, which is low grade. And so usually we give Tylenol or acetaminophen and that's it. And we just make sure it's better and then you're done. And we, keep an eye on it. Sometimes you might need a little bit of oxygen or you might need a little bit of fluids. But really that's what grade 1, 2 is. When that small number of patients that get grade 3 or higher, that's when we end up, saying, okay, we need to watch a little bit closely. Do we have to give medications to help your blood pressure come up? And really the nice thing with myeloma patients is that when we give something called tocilizumab, it's a drug we use for autoimmune disease, it actually helps knock these cytokines down and people do a lot better and they're kind of back to normal again. So for our patients at MD Anderson, we've actually had 2 patients in total go to the ICU and within 12 hours come right back out of the ICU because we give the toci and they're fine. So it's just going to a center that knows how to deal with this and that can act on it quickly. But the majority of patients do really well. And most people, the differences here are when that CRS happens. So in ide-cel, they have a lot more cells going in at first. So it usually happens at day 1 versus in cilta-cel, they have, smaller number of cells. So it takes them longer to get to that point. So usually it happens around 7 days. And then the other ones, obviously your counts can be low. So platelets, white count, neutropenia, we talked about the anemia, and we do have to transfuse patients sometimes. And usually after a month or two, most people are back to where they're okay and not needing any of that. But a few patients we have to watch for longer and potentially give more support for a few months. And then of course, ICANS, which is the neurotoxicity. Thankfully in myeloma, we don't see it like we do in lymphoma CAR T, but this can potentially lead to, some confusion or delirium, as we call it. And if it gets really bad, so grade 3, 4 is when people can potentially have seizures or go in a coma. And we don't really see that, which is good. We quickly give steroids to knock it down, and that usually reverses it completely. So for us, we have patients write a sentence every day, because if that sentence changes, that's an early indication something is happening. So we intervene so it doesn't get to that grade 3, 4. So we've learned a lot about how to intervene quickly to make sure we don't get to the really, bad levels of where it can. And so with that, I think this is my favorite slide, because this really shows, for my patients, when we talk about patient experience and why people, are so into CAR T, that one dose of cells, right? You do get 3 days of chemo just to knock your immune system down, so that when these cells come in, your immune system doesn't just clear it all out. We need a little bit of time for those cells to find the myeloma, do what they do, and then your cells come up and kind of kill them off. And so this is one of my patients who's a high school teacher, and one of my patients who's a nursing supervisor. And both of them were working through COVID, at school and all this, and their myeloma just took a turn for the worse. And both of them were able to get onto a CAR T trial, and actually are both doing fantastically. They've both gone back to work, and despite COVID, et cetera, and are actually doing really, really well. So for my patients, what they tell me again and again, can I go on another CAR T trial? Because it gave me that chemo-free period. I felt so great. I was able to do the things I want to do. And hearing that, I can't not hear that. So this is why I think for me, it's such a big deal to be able to do something like this. And then, like I said, we do have the initial approval already for idacaptogene. So we're working on getting more patient standard of care CAR T now, which is a whole nother ball game. And there have been some, bumpy road along the way, but with potentially cilta-cel hopefully becoming approved also in the end of this year. Their FDA approval date is the end of November. I'm hoping by January, we'll be able to, get more and more patients on. So then going into the bispecifics, and really the picture on the side just shows you that there's two ways bispecifics, there's multiple ways bispecifics work, but there's the bispecific antibody, meaning that you have this antibody that attracts the T cell by something called CD3. It kind of brings that over. And then the BCMA, it brings the myeloma cell over. And I call this the handcuffs, right? It's handcuffing both of them, bringing them next to each other so that T cell can kill that myeloma. So this is sort of an indirect way of getting that T cell there. And then you have a T cell engager, which is more like magnets, right? It actually puts both of them together and puts a little fire under that T cell and kills that myeloma cell. So, there's been multiple studies that have been looked at, and I'm just showing kind of the response rates of the 3 that were presented at our big oncology conference, ASCO in June. So elranatamab, overall response rate, 70%. Teclistamab, overall response rate, 65%. So these are also still 2 to 3 times what it has been for most other drugs. And those 2 are both against BCMA. But the other really cool thing is that we're starting to find new flags or antigens for myeloma, and GPRC5D is just one of them, but we have multiple other ones too. And so one of those bispecifics was actually talquetamab against this new antigen. And once again, overall response rate, 70%. And so, these are fantastic for single agent. And so I'm not going to read this, but you'll get these. But these are some of the highest overall response rates we've ever seen for relapse refractory myeloma patients. And really our question for the next 5 to 10 years is, how do we sequence, as we talked about with BCMA specifically, or how do we combine autologous transplant, BCMA CAR T, and or bispecifics? Can we give CAR Ts and then a bispecific later to help them work better? Maybe. There's lots of things that we are wanting to do, and hopefully we'll have trials for soon. And then, of course, how do we cure patients with CAR T? If we can get these responses like this in very relapse refractory patients, how can we finally cure some of our patients? In lymphoma, we can do it. In leukemia, we can do it. So now we got to get smarter about how can we do it for myeloma patients. And that's really what, looking at these mechanisms of response and resistance to help us get smarter about how to fix that bone marrow for once and for all. And so, the other patients that really, we talked a little bit about the unmet need, those who don't have access to appropriate healthcare clinical trials, we really need to do better here and, make sure that all patients get access to care. High risk disease, plasma cell leukemia, they do CAR T and bispecifics seem to work better for them. But if their disease is so bad, they can't even get to the trial, well, we've lost those patients. So we need to figure out a way to get patients newer agents like this earlier. Relapsed refractory disease for patients who are really aggressive. Kidney failure, we talked about. If there are people with CNS or, involvement of their brain or their the fluid in their around their brain and spine, which is hard to treat. And then, of course, frail patients with increased comorbidities. These are the kind of the things that we really need to work on, and hopefully we'll eventually be able to treat, all patients and hopefully cure, like I said. And then just a quick plug on the different trials we have at MD Anderson for CAR T and bispecifics. Really, the autologous CAR T, we have to collect your own cells. There's a bridging period, about 3 to 4 weeks, and it takes a little bit of time. But most of these trials are for patients who are relapsed or refractory, at least 3 lines of therapy. But as you see up here, we've actually been doing trials in earlier lines to see if that works better. And right now, KarMMa-3 has already just finished recently, so we'll hopefully have data on that soon. KarMMa-2 is finished enrollment for the most part. And KarMMa-4 is going to look at upfront patients. So patients who are high risk that we actually give CAR T to instead of stem cell transplant. So I'm really excited about that trial as well. And then KarMMa-7, we're actually combining things. So as we said, we're trying to become smarter about it. We're combining different therapies with CAR T, like CELMoDs that we didn't really talk about, but that could potentially improve CAR T, or ways to make that BCMA work better. So that's a really exciting trial. And then we have, of course, off the shelf. So people who can't wait for those 4 weeks to make their own cells, because we have that as a problem. We have allo CAR T trials where you're using someone else's T cells that might be healthier because they haven't seen cancer or drugs. And there's different trials we have for that, including one that's against CS1, which is a different antigen. And then we have, of course, our T cell engagers that are, BCMA mostly right now, but we have others that are coming down the line for other antigens. So, with that, I just wanted to thank our patients, caregivers, and research teams, because we have so many therapies because of all these trials, right? And because of our patients and our caregivers who actually volunteer to go on these trials. And I will say 7 years ago when I was a fellow, we didn't have CD38 monoclonal antibodies and all these things. Really, we had novel therapies like bortezomib and, lenalidomide, but all these other things were in trials still. And so at this point, we use them, like water for our myeloma patients because they work so well. And so really, it's because of them that we've even gotten to this point where we have options to individualize treatment.
I think that we're looking at a future where BCMA therapies are going to be brought in earlier and earlier. And we will, after even just 2 lines of therapy, potentially have 4 or 5 different classes of refractoriness, which really is going to, put us at a decision point of what is the best next thing to give. And that's why agents like selinexor or melflufen or these other ones and anything else that we will come up with hopefully are so important. And that's not to say that they're only reserved for those time points, but the point is the more we have, the better, and the more diverse the mechanisms get, the more hope we have that these resistant mechanisms that Dr. Patel mentioned can be circumvented. So I'll probably stop there. There will be many other things to talk about, but these are very exciting times for myeloma.
Thank you for that. I appreciate that, Dr. Baljevic. And Dr. Patel, any last words from you as well so the takeaways for patients and caregivers?
Yeah, no, I think that it's a good thing that we have so many therapies. A lot of people get overwhelmed and they're like, how does anyone pick? And are they going to pick the right thing for me? But I think the more options you have, yes, you have to go to someone that knows myeloma, or at least help your doctor with your treatment. But that's who can actually help to make sure you get that best therapy. It's not an algorithm, right? And I think this is what myeloma has been for the last decade or two, is that when we get new therapies in the relapse refractory patients who are phase 1, phase 2 trials, usually the phase 3 trials are earlier and earlier, and then that tells us we can use it sooner. And in the end, it's still about using your best therapies up front, because that usually gives you that longest hibernation period and hopefully one day cure, right? If we can cure it, we're hoping we can cure it at the smoldering stage for patients, right? Before they ever get any kind of organ involvement. So that's sort of why it's set up the way it is. But I think that, I wouldn't be afraid that by using a quadruplet or doing BCMA early, that you're going to run out of options either. That's not the case. The goal here is, how can we give you chemo-free period, or how do we reduce therapy? So the likelihood is that even if we give you a quadruplet plus a BCMA, once you get to that best response, we're probably going to take things off, just like we do now. Right. And the goal is that you actually get a much longer time in that hibernation because of all these things, and that more people get a better response. Really, it's to make sure that everybody gets that response, as to why we change things around. So yes, I don't think I'll ever be able to retire. I was hoping I get to retire in like 5 years, but I don't think that's happening. But I do think that this is huge, and why we're getting so many more people interested in myeloma as physicians and researchers, because that puzzle, we're sort of putting it together, and sometimes the more you delve, the more you learn, and it becomes a little bit more confusing at times. But once again, gives us more opportunity to actually treat all patients with myeloma. So it's really, really exciting. And I think the talk today was fantastic with Mohammed, my co-fellow from before. And I'm so happy to be able to do this today with both of you.
Thank you both so much for spending so much time with us in setting the landscape. I know there was a lot to get through. And I think, thank you for not retiring yet, Dr. Patel. And I hope you too, Dr. Valjavich, having people like you who not only clearly, know what you're doing in the research, on the research side, but really care about the patients, because I've heard it come up several times throughout the entire discussion, about the patient experience, about the impact, Dr. Valjavich talking about financials or financial toxicity. I mean, these are so important. So we appreciate from patients and caregivers when doctors are holistic in their approach. So thank you so much. And for audience questions not answered today, we will try and do a follow-up. And yes, Dr. Valjavich.
And I'm sorry, I don't mean to say the last word, but I just wanted to use this opportunity, Stephanie, to thank you for the work that you do. First, I believe I speak for both Krina and myself when I say that we're grateful that hopefully you've done well and that you're here with us today, because as you pointed out, you're a cancer survivor as well. And two, these types of events and education materials are so important and so beneficial. We hear it all the time from our patients, and it gives us a valuable opportunity to engage and to try to address any of the additional questions that, as Krina mentioned, we have limited time in clinics with our patients, and it's just sometimes not enough to speak about everything that we think is important. And I know, and I'm sure your viewers will agree, as we all feel in this field, that these types of education efforts and materials that are available to patients that stimulate them to think about their disease, that stimulate them to think about reaching out to specialists at least once during their disease course, are really important, because they are pushing the boundaries forward and forward. And we know that there's data that clearly talks about improved outcomes when we all come together. I just wanted to use this opportunity to thank you for that work, and it was a pleasure to speak with both of you today.
Oh, thank you so much. I feel very grateful to do this work. And as we know, a lot of people in this country don't have access to larger healthcare providers and academic centers, and to people like you. So we're going to push this out as much as possible so we can reach as many people as possible. And for the full talk and for both stories, we will post it at thepatientstory.com, where you'll find human answers to your cancer questions. Thank you so much.